Okay, hi everyone. I think you came to the right place. So welcome to Cell Biology and Sustainability by the Numbers, and all sorts of other things as well by the numbers. And it's going to be quite an adventure for me, and I think for you as well. And let me start by, just big picture. Why do I think it's good to be in this course? So I think, as scientists, but in general as people, we want to understand the world. The world is super complicated. There's a lot to try and understand. And a real good and effective tool is numbers in order to try and understand. And that probably doesn't surprise you. It's something, we see all around us all the time. But I have to say that I think for most people, numbers in the way they're being used is often just like a sort of a decorations. Like you see numbers and it's a nice thing to, to say that you have or something like that. But really getting all the potential from quantitative understanding requires much more. It requires all sorts of numeracy that we'll be talking about. It requires some hands-on experience. It requires context and all sorts of tricks and of the trade. And this course is all about how to develop this skill through practice and through lots of examples. And I think from previous years, I think you'll find it interesting, or at least I hope. And so this is the general motivation for the whole course. And I hope you'll also find that it's useful for many other regimes. So even though we'll take examples from biology and sustainability and today from the Olympic Games and some other stuff, it's really translatable to many other things. So, and this you'll also have the opportunity in the course to bring your examples, for example, to the final exam to apply it to whatever you'll find interesting. Now, usually when doing such talking, there's so much talking, everything seems too abstract. So it's usually good to go to some concrete examples pretty fast. So let's first of all see, in terms of we're like 2022, why still would we have courses like in regular format and with people coming and meeting in person? Any suggestions of why it might still be useful? Or not? Sure.
People get lost in Zooms and stuff like that. And if you want a course that you are able to participate and talk and share your experience, you can't really do this in large numbers by Zoom.
Excellent. So this enables you to participate and not being lost. Great. Any more suggestions?
It's less boring.
Less boring. I'll take it as a compliment. Less boring. At least so far. Okay. Yes?
More social.
More social. I love it. Which is, by the way, interesting, right? Because, I guess everybody here are, coming from the scientific discipline. So often we have these notions or paradigm in our head that like answers to such, utility questions of why do something, just more social is like, it seems like it's not like a good scientific answer, right? We want something more of like, a good, but actually, something being social is so critical for us having, for example, motivation. And motivation is clearly something that's relevant in order to be able to learn effectively, etc.
To keep this direction, later we discover in the industry that there is a iterative process of technical and personal abilities, so they push one another.
Okay, so iterative, all sorts of stuff. Yes?
We probably get a better experience from you. Like you give us a better lesson that you have people to talk to versus just talking to a screen.
Yeah, so experience, which means that you were mentioning me as something that will give you some experience that hopefully would happen. But I think you'll also see the power of the group and learning as a group, as well as some of the things you'll be doing, and I hope you'll experience it starting today, like say working in pairs, learning from each other, and that's what I wanted to say. Okay, anything further? Yeah?
A lot more engaging.
Engagement. Okay, so there's more to be said, but so what's the alternative to all this? Right now, there's like a lot of talk about something as an alternative, which actually I connect to and like very much. These, massive online courses. You might have heard about it, edX, Coursera, other stuff, things like that, which have a lot of potential. You might have heard about it. It's like you can learn whatever you want from the best teachers in the world, not just those that are here with you at the Weizmann Institute. You can do it anytime, any place, and I think that's very promising. It's so promising that I was considering whether I should do this course in that manner. And so I started looking at this, and we're ready to get our first hands-on experience. So once I did that, I participated in the course, and then I got an email back at the end of the year telling me how much is being done in such courses in one of the leading platforms in the world. And you have what they sent me already in your inbox. So if you take your phone out, those who do not have the phone or don't...
It's the Weizmann account.
So just look at your Weizmann account. You have the first hands-on experience with by the numbers. Those who do not have reception or something like that, you can just move and sit next to somebody who does. So look at it for 2 minutes and tell me. So this is like end of the year description of what happens in these online learning, and I'll be happy to get any thoughts once you looked at it a bit. Okay, so come back for a second. So first of all, I'll say, all of this is it's not about speed. So, it's more of at the Weizmann Institute, we're looking into depth. So, but sometimes just in order to, we'll move from one thing to another quickly. Don't worry about how quickly, you reacted, somebody else reacted, etc. Okay, so who noticed something they want to share, just generally? Yeah?
There are very big numbers in the email.
Yeah, so very big numbers.
When you look at them, it's like less than 10 minutes watched per person.
Okay, so let's look at it for a second. So just to be clear, what we have here is the report that is sending probably to how many? So let's see, what do they write there? Can you remind me just generally? The numbers.
33.6 million people enrolled.
30 something? 30 what?
33.6
0.6 million what?
enrollments.
Enrollments. Okay, so they have 30 million people who enrolled in courses. I think that's pretty impressive, right? I know that also, like, you see this actually, I think they write it not this way, but they write it like, it's like with what is like 8 digits, which even looks more impressive, right? They know to the last person out of 33 million, that's like, the population of I don't know what country. So you have like a whole country just studying with them online, freely available, super impressive, right? And usually you continue from there, and it all seems quite impressive. So what else do we have there?
118 million problem sets.
118 million problem sets. I think they have something about minutes, right?
Yeah.
Yeah.
What is it there?
229 million minutes of video.
229 million minutes. And okay, what else? There's something about the number of courses.
1,300.
How many?
1,300.
1,300 courses. Okay, and all that is written like with full, very, very large numbers, which looks really impressive. Okay, but now somebody noticed something else, right? Yep.
if you divide the amount of minutes that people watch by the amount of courses that people enroll, you see that they watched an average of about 8 minutes per 8 or 6, 4.
Great. So let's take the number of minutes, and we divide it by the number of enrollments. Enrollments is number of students, right? Number of minutes to number of students. So we have about 30 million. No, excuse me. We have about 200 million divided by 30 million. So that's roughly 10 minutes per student. Okay, so now we could do a whole discussion whether it's actually 7 or 10. As you'll see, we're developing like a new algebra here, in which 200 divided by 30 is like 10. But you know, with the grades, I'll try to be more accurate. But with other things, we try to give the level of precision or accuracy, or I never remember the difference between them, in such that it would be appropriate. And here I don't care so much. You can see we're talking about the fact that even though they have that many students enrolling, per student, not so much. Okay, so I think that's one thing to notice. Anything else that you can infer here? So you know, some people could say, yeah.
They don't really give information that you care about, right? Like, it's more interesting to know, like, oh, maybe of those courses that have 1,300 courses, are there 3 that is most of the enrollment? You know, of those problem sets that are attempted, how many are complete? Like, it's not.
Right, right. Okay, so that relates now, for example, we could look at the number of courses, and now I guess like the question is, okay, you have all sorts of numbers. You can use them as decorations. It's not just like really cool stuff. I know the numbers, and like it's, you know, data science, there's lots of that and everything. But then you look a bit more deep and say, okay, what do I care? What is the question I really care about? So here's one concrete example. You know, if I'm a student going there, okay, what, you know, or if I'm funding this or donating. Actually, they were asking me to donate for this, which is actually very nice. They're saying you enjoyed it, we gave it to you for free, now you donate to this, which I connect to. But then you want to see, okay, what's the, so you see, each one is spending 10 minutes. Okay, strange. Now you can also look from the perspective of, to make such a course, it's really a lot of time, a lot of effort. Like you have to sit inside, in front of a camera, prepare anything like really well. It's a lot of work. And then you ask yourself, okay, what am I going to, as a teacher, would that achieve like the notion? What is the notion? I'll do that, and then everybody, in all the countries in Africa, Asia, Americas, Europe, would enjoy me as their teacher, and it would be super effective. Okay, so we could try now infer something about it, because what do we see here? Let's take the number of minutes per course. So what do we have here? It's the same thing. 2 times 10 to the 8 minutes, divided by about 10 to the 3 courses. So that's about 2 times 10 to the 5. So far, so good? Okay, that's also a pretty impressive number, right? It's like 200,000 minutes. Minutes per course. So that's also something that's actually usually useful. If you just leave it like, with units, it's good to start, like we usually don't think about teaching in units of minutes, right? We're thinking more of like, say, over a semester or something like that, right? You want to know how many like effective students do I have here, okay? So then I would think about, okay, let's think about, you guys coming to a course. How long do you spend in a course? Okay, so how do we infer that? You're saying, okay, it's like the number of lectures, oops, which is roughly what? 14, 13, 12, maybe we'll have all sorts of festivities, new, I don't know what that, so let's make it 10. Okay, which is usually the answer to almost any question I'm asking. Okay, number of minutes
100
per lecture. So obviously it's also 10, but just 10 to the 2. Okay, 100. It's 60, right? Or not 60. Okay, if it's 2 hours long, then it's 120. Okay, let's make it 100. Okay, so that comes to the answer that you heard here. Okay, so together it's like about 1,000 minutes per. What should we call it?
Per student per.
Yeah, per real student. Okay. So it's about 10 to the 3 minutes per real student in a course. Real course. Okay, which that means that like if I take from here, that means that I have effectively about 200, let's call it full enrollments. That's also, it's not bad, but
Compared to the real number, compared to the number they give you, which is 25,000 per course.
right. So so as noted here, the notion that people have about, that okay, you'll work hard, prepare a course like super high quality, and then you'll have like these unimaginable classes of, tens of thousands of people, like really huge effect. 200, well, that's not very far from what people, are having in usual classes. And we have to think about who are these students. It's like they have like pretty strong attention deficit, right? They're coming for like usually 10 minutes and then they're gone. So you have maybe some, but they're hardly with you. So effectively, it's not so much if you look at what you're getting. And then there's the other point that was mentioned here. We don't know anything about the distribution of this. So probably it's not that like everything is homogeneous that everybody. It's probably there's like a course in machine learning from Stanford that 100,000 people are watching quite a lot. And then a very large fraction, are getting a much smaller number of minutes a percent. Okay, I could do much more with this, but I don't have that much time. But the hope is that you got the message of what you can get by just not looking at numbers just as decorations, but like looking more deeply and with some playing around with them, putting them in context, etc., which in my perspective is like a sixth sense. Right? We have the regular senses, but now with being like really quantitative, you develop a new sense, which sounds like crazy. But yes, I believe it. Or at least, I'm making that claim, and you can test me along the semester whether you feel that that's justified. Okay? Comments, thoughts? Okay, we're making progress here. Yes?
We came to a number here of full enrollment students, and the website gave you a number of like enrolled students per course. If you just calculated the students per the number divided by the number of courses, what's the more real number here? Like, could you gain some insight just based on the calculation that you did about how many students you actually have in your course, or do you understand that?
So let's say you're saying another way to play around with the numbers is to, for example, divide, if I got correctly, say the number of enrollments, 30 million, by the number of courses, 1,000, which would mean that that's about 30,000 people per course, or 20,000, or the like. Wonderful. Okay, so you're asking whether this also gives some information.
We just did a calculation, which basically says I have 200 fully enrolled students based on the numbers we did here. Does that add some sort of insight to the number that they gave us? Which one would be more real as a number?
Excellent. Okay, so the question is, which one is more real? So I think it's a great point to notice that, our brains are so like circuited, to have like a right answer and the wrong answer, or like this is the useful thing and this is useless. And actually, the world is more complicated. It's not black and white. This is useful for something, and that is useful for some other things. And therefore, there's no, I think the kind of like this calculation that we did, I think is the more useful thing, because telling you that there's 30,000 students enrolled on average per course, where you don't know for how long are they spending with you, and then it's not so useful. So I would say that's much less useful, but it is also saying something. Good. Okay. Everything we're doing is in this course, you'll see I'm trying to teach through examples. Okay, I think this kind of active learning is part of what makes for effective teaching, and you'll need to participate. I hope you're fine with that. And it's also part of what's known as this kind of thinking, and is what's known as Fermi problems. Anybody knows anything about either Fermi or Fermi problems? Yes?
Is the problem with the fact that aliens exist but we can't find them?
Okay,
Or intelligent beings exist.
okay, so I think that's the
Word smart.
That's the Fermi paradox.
that's the Fermi paradox, exactly. Also, well done. So this guy Fermi, he did quite a lot, right? He was like super smart, very creative, did all sorts of things. He started the things that led to the atomic bomb afterwards, all sorts of things that had quite some repercussions. You're talking about something else. Fermi problems. What?
Lots of numbers.
Lots of numbers there. That's true because yes.
How many piano tuners are there in Chicago?
Right, so that's a classic example by Fermi. So he was working in the University of Chicago, I think after he fled Europe. And what he was doing there in the 30s is teaching all sorts of things, doing research in physics, but also giving, for example, in his courses, the example, okay, who can tell me how many piano tuners there are in the city of Chicago where they were living, or where he was teaching? And so most people did not know, but he knew that. He wasn't expecting them to know how many piano tuners there are. What he wanted to see, whether they could infer it for themselves. How can you do such a thing?
How many pianos? Start with that.
Okay, how many pianos?
How many people in Chicago?
How many people?
Population.
Yes.
What's the average in the world per city?
Yeah, so you can start and be quantitative about it. And I want, you could play around with yourself. But that's a classic example, and there's many other types of such problems. It basically, you break it down into, say, for example, okay, how many people are in Chicago? What fraction out of them plays the piano? A person that has a piano, every how long does it need to tune its piano? Tuning a piano, how long does it take? And then I already know how many hours of piano tuning I need for the city of Chicago per year. Okay, how many hours does a piano tuner works per year? I divide the two, I get an estimate of the number of piano tuners. Something along those lines. I wouldn't be surprised if some of you decide after you finish at the Weizmann Institute to go and go to the high-tech industry or something like that. And there I wouldn't be surprised if very often people in the interview would try and ask you some crazy question, I don't know. I've heard that in Google sometimes they ask you, okay, you have like a truck and you fill it with golf balls or ping pong balls, how many ping pong balls you can fit in there? Or you want to go and take all the lane from Tel Aviv to Jerusalem and paint the white thing you do on the road to separate the lanes. Okay, how many buckets of paint do you need? All of those questions are types of Fermi problems where you want to see how well can you think on your feet, how quantitative can you be, can you take a problem and break it down. So if you want, that's the sources of the kind of things we're doing. It's something also very common in engineering and physics and what's known as back of the envelope calculations. Okay, like what can you do by just, back of an envelope? This is kind of an envelope, what we have here on the whiteboard. So let's have a concrete, anybody wants to suggest, cool questions like that they've heard while I'm preparing for the next thing? So if you ever heard about such a problem or have something you want to raise as a challenge, it's a good time.
For my bachelor's, they asked me how many stairs we have in the Eiffel Tower.
How many stairs in the Eiffel Tower? Good.
I heard people being asked how many windows there's in Tel Aviv.
How many windows in Tel Aviv? I've heard it about Manhattan. Good. Well, let's see how are we doing so far. I see some participation. Less boring, you decide. More social, we'll move to that. Iterative will happen. Experience and engagement. Good. Any more examples?
Yeah, so the Fermi paradox is the how much intelligent life should there be in the universe based off of the...
Right, so the Fermi paradox, how many other civilizations or planets are there out there? Which is like this is like at the top level of trying, this is tougher than how many windows there are in Tel Aviv with different implications, but it is a type in a way of such Fermi problem. More examples people want to share?
There's this like debate.
What?
There's a debate on social media if there are more doors or wheels or something.
Okay, so more doors or more wheels in the world. Okay, that's also kind of related. Also, that's like, so it's like two separate, if you want, it's like two separate Fermi problems that you can combine together. Okay, so here's my my next challenge for you. Anybody likes to watch the Olympic Games? Okay, so in the Olympic Games, one of the most popular things is the 100-meter dash, right? You're like running. How long does it take to run 100 meters if you're at the Olympic Games? You see, everything is 10. It's like really like, okay. But now they also have this thing that they have to decide if, if I'm now sitting, standing there and I have the pistol of the judge shooting, and I have to start running. They want to see that I'm not tricking them, right? Because if it's 10 seconds, every second or every millisecond counts. So therefore, they have a threshold. And they're saying if you start running because you were just guessing when they'll shoot or something, then you're disqualified. Or actually, it's more complicated. The first time they say, they cancel. The second time they disqualify you. Okay, now my question to you, we need to help the Olympic Games and decide what's the threshold, what's the time at which, if you started running, this is okay. Like you really, it's fair. You actually had a chance. Like it's not that you were guessing and like or something like that. Question is clear? Okay, so what should be the threshold?
Threshold from the moment of initiation of the sound, or
Exactly, from the time they shoot the gun,
from the start of the.
from the shoot of the gun until a person starts running. If it's what's the threshold? That if it's less than that they started running, then it means that they were bluffing.
Depends on the distance between
Whoa, just 1 second. Let me. Okay, let me first of all, to make it interesting, it's good to have, when you're trying to approach a question, to first have some preconception, like try to have, put your money somewhere. So let's put a few options. A, let's make it 1 millisecond. B, 10 milliseconds. C, 100 milliseconds. D, 1 second. Is it clear what I'm asking? Okay, who's voting for A, 1 millisecond? 1. Who's voting for B, 10 milliseconds? Oh, it's hard counting this. I would say 2/3. Who's voting for C? The other third. Who's voting for 1 second? Good. Okay, 1 second does sound strange. I agree. This would be like slow. It would be like a slow motion hand on bush. Let's go. Something like that. Okay, so that doesn't make much sense. But now we seem to be, now the question is about the other options. Okay, how would we approach such a question?
If someone wins by 1 millisecond, can they even measure it? Is it sensitive enough?
That's a good question. Anybody knows? Like if it's like 1 millisecond, can you tell the difference in terms of
Yes, you can.
I think they can, yeah. I think it's interesting to see exactly the resolution, but I think they could do better even. Okay, so we need to take this, yes?
Probably you would want to know what the reaction time for a person is. Like, what's the physically fastest time?
Okay, so we have issue of reaction time. What else?
speed of sound.
Speed of sound.
The distance between the pistol and the.
Distance between pistol and the person. Okay, so there's several options here. Okay, that's what makes life complicated and science complicated. There's all sorts of things playing around. Some of them, like reaction time, is pretty complicated by itself. What do we do when we have complicated problems? We just despair.
Like everything ones and zeros.
Now, so what we also, another option is to do this thing, divide and conquer. Oh, okay, we did the Fermi. We're at the Olympics. Now we do divide and conquer. Okay? What is divide and conquer? You try to start and break down the problem. So we have, you'll see I'm very good at making paintings. So we have the judge, and they're shooting the pistol. Okay? And now we have the people here. that are waiting to run. Okay, so now what needs to happen? So we had things about the speed of, so there's like one step that this has to reach the ear. This would not reach the ear this way. So from the gun, it needs to, you have this as like step A. What needs to happen next? So there's something that needs to happen in the brain. I don't know, thinking, deciding, whatever. This would be like part B. Then you have to start moving, right? So you have to have the action potential from this going here. That would be step C. Okay, and maybe there's other things about there's something in the muscle here, and I don't know what would take until they actually start moving. Step D.
Can I just ask something? So C, what you do as C, so the action potential, it's something that, like I'm involved in this business of running coaching, and this is something that is actually like, this is what separates the average person from Olympic guys. So if you don't train for this specific movement, so the time it takes for the action potential to move from the brain to your legs is like much higher than what people would think, like several days actually biologically. So it's a movement that the body needs to develop. So what I'm pointing to is that actually this should be very much different from.
Okay. Okay.
So you need to average. Let's say you need to average.
Okay. So there's something very nice here. We have in the audience, somebody who like really understands a lot in this. Okay, which is really cool. And let's move a bit further, and then let's see how it connects. It also repeats itself in other problems where there's always this, let's say, tension between like knowing enough about the problem, like knowing a lot about the problem, like being the expert, and trying to get an answer. Because I guess if I'm asking you, it's not so easy to say, okay, so how do I decide that on this, based on this? It's complicated. We could talk about it. But anyway, let's move on. Just 1 second. We have a lot to achieve today. Okay, so we could take each of such parts. For example, we could say, okay, how much does A take, right? How would you approach that? Yeah, so we have the distance, we have the speed of sound. That would give us some delay, right? There's something strange with this, no? What? Yeah, so the distance, it depends where you're standing in comparison to the judge. It's not so fair, right? And you can make the calculation. It's like 300 meters per second, the speed of sound. And if there's a distance of, I don't know, like 10 meters between them, or even 3 meters, it would make like 10 milliseconds. And maybe you'll be winning just because of that. So what do you do?
The distance between the different
Put them even distances from the judge.
Yeah, so even distance from the judge, that means it has to be on the lane. And then they just run at him. Okay, so it could be at the behind. Actually, so that's kind of like what they do. You know, the way it works now, the pistol, it actually makes no sound. It makes no sound. It's connected electronically to loudspeakers close to the butt. Like where, they're like this. They have loudspeakers in the behind. And like it comes all synchronized at the same time. Olympic Games, they want it to be as fair as possible.
They do these things in Olympics, or you just?
No, I didn't make it up, even if I tried to, I wouldn't be able to make this up, There's no chance I could make this up.
What's wrong with lights? Race cars use lights.
Yeah. So anyway, so that's how they do it. So actually, it turns out this is not so critical, although you can talk about the time it moves from here to there. Okay, we could break it down. I don't have time for all this. Let's jump directly. So what happens in the brain is always a mystery to me, so I won't get into that now. Let's talk about step C. Okay, what do we know in order to so we did a divide and conquer, right? We broke it down into different aspects. Now we took one component, we want to dig more deeply into it. What do we need to know for that? Okay. If like more from like a physical basis, biological basis, what needs to happen here? Yeah, so there's an action potential that has to run on neurons from the brain to the leg, to the muscle. Okay, so what's the overall distance? Yeah, so we have about 2 meters, right? These guys are usually tall. Okay, so now what are we still missing?
You have this
The speed.
motor proteins that takes it, but as I said,
So we had two answers here, right? One was the speed, one was the motors of the this or that. It's like biology is full of details. What you will try to do in the course, in some cases, is just to say, I don't know the details. I want effectively to just think about the speed. It's true that it's based on like synapses and dynadine and dynein and myosins and actins and all sorts of things. And it's myelinated, it's not, it depends whether it's this neuron, that neuron, whatever neuron. But in a way, we could just think of effectively what is the speed. Okay? Good. So if I know the distance already, I know L. Now I only need to know the speed. Anybody knows something about the speed of action potentials?
Very close to the light speed.
Okay, light speed, that's pretty quick. So light speed is, it's not the light speed. I don't know. It's not the light speed. Okay, so.
We're talking about two different speeds, like the electrical one and the chemical one.
Yeah, so there is electricity in there, and that, intuitively connects to the speed line, but there's things that need to, ions need to go here, need to go there. It's a combination of chemical and all sorts of stuff. I'm guessing you don't know the speed of neurons. What?
For Google, 100 meters a second.
100 meters per second.
It's a nice number for you too, so.
Okay, so it also, as always, it also depends somewhat, but it's somewhere between 10 to 100 meters per second. I want to show you something else as well. In many times, you won't know, all the numbers needed, but I think there's something nice in the fact that you'll find that, like, from very few numbers, you can infer a lot of numbers. Okay, so in this case, we need to know something like the speed of action potentials, and then it could connect to many things, many other questions. So even if you don't know that number, maybe now you know, or maybe Google knows that, or, okay. So now if I have 2 meters, and let's say with 100 meters per second,
Another thing that you can do is to estimate the time directly without the...
Just 1 second. There's all sorts of ideas of improvements. I won't have time for everything. I'm sorry that I'll stop here. So I can infer a time based on the distance divided by the velocity. This gives me about, let's do it properly, 2 divided by 100. So that's about 0.02, which is 20 milliseconds, right? Okay, so this is like a pretty fast speed. There's all sorts of other things happening here. What I think becomes pretty clear from this is that this could not be the answer. And actually, this could also not be the answer. Because like this 20 milliseconds, that's like we took, it's only the action potential without anything, no motors, synapses, other responses, not anything that's happening in B, anything that's happening in D. All of that is not included. And that enables us to see that like the answer among these should be 100 milliseconds, which, by the way, I think is the right number. And all those, as you mentioned, people are preparing for it, so you can see they actually have statistics for the people running the 100-meter dash. And you see the really top athletes are achieving somewhere between 110 to 120 milliseconds. These are like the top athletes. Good. So what did we learn from this? I think we learned the divide and conquer, how we break down a problem. We learned that, like, okay, we were missing some numbers. I'm not expecting you to know all the numbers, but from, kind of like one kind of number, about just, speed of action potential, we can infer things about, Olympic Games, and believe me, all sorts of other things you can infer about human behavior, physiology, something like that. And I wonder, like, does that raise any other questions? Let's say, okay, we've gone through that. Does that, anything else that comes once you played around with this? Or we went through this example quickly. Take your time to see if there's anything that it makes you curious about.
If you
Yeah?
Shorter people are faster.
Okay, so maybe shorter people have an advantage here. Good.
So it's like 100 milliseconds from the time they shoot it, like all this you guess, and they're just like.
So the threshold given by the Olympic Games is 100 milliseconds. That's what they decided. After shooting, yeah. After shooting and before they were allowed to start running. And they check that by the fact that there are sensors on the whatever they're standing, and they see if you lift your leg from the sensor.
Yeah. More generally, like what is the variation of the parameters that you used? Like just interesting to see how can you affect this number. What would give you?
Okay, so for example, you're saying, could there be? I'm paraphrasing from that. Could there be variation in the speed, let's say, between people? Can you, for example, by training, can you make that, this running faster, or how much does it vary among different people? How long does this take? This thing in the brain. How fast can you be here? What happens in step D in terms of like, okay, you got the action potential. Now you need to have the muscles doing something. But then there's all sorts of other things. We were talking here about somebody shooting and then what happens. But for example, when you're watching a movie, right, and you're seeing something happening, like continuous. It's not continuous, right? They're putting images. How many images?
24 images.
Yeah, something like 24 images per second. So you can divide from that. It's about 40 milliseconds between one and the other. Is that connected to what we did? Is there so all sorts of things that you could take based on like, doing something that like now give you new questions, new understanding, deeper understanding into all sorts of things. Okay.
Can I ask a question?
Sure.
So we arrived at 100 milliseconds for just doing this calculation, and the Olympic Games also arrived at 100 milliseconds. Is there a reason we arrived at this round number that they also arrived at? Like, is this round? Because this is like a very comfortable number for us, but they arrived at the same number. Is there some truth to this number? Again, like, would it 0.932 be a more correct number?
Okay, excellent question. So I think so the question is whether we have here, like, how come that it's 100 milliseconds in this, like, I was using this very convenient, comfortable way to write numbers, but is that also is that how the Olympic Games decided to do it that way? Or is it a coincidence? Or I think in this case there is some coincidence here. I don't think the Olympic Games, if the response time was 200 milliseconds, I don't think they would have left it 100. Then they would have made it 180, I don't know what. Or if it was 70, they wouldn't stay with 100, they would put it 50. So on the one hand, there's an advantage to having like round numbers, and therefore they were happy probably with 100 milliseconds. But it's sometimes, okay, it's a question also of like how accurate you want to be. Sometimes, like in things like that, you might even try to push it into be more accurate, like to choose more digits. We'll talk about it later in the course. In this case, it comes out pretty comfortably and nicely. But I think what we were able to do is like to differentiate between very different ranges. I think that's the key of what we could achieve here. Okay. Let's move to another example. As you'll see, the way the course moves is often between examples, and I think from that you'll be able to generalize with time. So let's think of something else, and that's the okay. What is the number of proteins, protein molecules inside a an E. coli cell. Okay, E. coli cell, for those who don't know them, are these nice model organisms, bacteria, used to live in our gut and now living in labs all around the world. And now let's give us some options. A, is it somewhere between 10 to the 3 and 10 to the 5? B, 10 to the 5, 10 to the 7. C, 10 to the 7 to 10 to the 9. Or D, 10 to the 9 to 10 to the 11. Is the question clear? Okay, so we're not talking about the number of types of proteins. Anybody knows the answer to that? How many types of proteins there are in a bacterium? Right, a few thousands. We talk about the number of copies, like, I'm counting one after the other, all the different proteins. And we want to know the rough order of magnitude, right? This is called orders of magnitude estimates. Okay, if you understood the question, now you need to vote, like we do in Israel all the time. Okay, who's voting for A, 10 to the 3 to 10 to the 5? Only me. Okay, who's voting for 10 to the 5 to 10 to the 7? I'd say 1/3. Who's voting 10 to the 7 to 10 to the 9? Another 1/3. Who's voting for 10 to the 9 to 10 to the 11? Kind of 1/5, I don't know, and I lost something along the way in the fractions. Okay, so we see we have quite a range here. And now the question is, that's good. It's tough to develop intuition for big numbers, right? These are like big numbers. It's even worse. It's big numbers with small organisms, right? It's like the worst it could be. Like we have no intuition in what it is to be a bacterium. None of us has been a bacterium for the past 3 billion years or something. You have been, but just it's been a while since then. So we forgot. And at the same time, these are big numbers. So also we don't have a great intuition into what it means, 10 to the 3, 10 to the 7, 10 to the 11. Both are really big. So we don't really have great intuition to that. So the question is, how can we do the same thing of kind of like what we did here to try and divide and conquer the problem? Okay, so this is what I suggest we do next. So let's take like 5 minutes. You can do the following. Like, I think it's best, if you want to work by yourself, but it's good also to work in pairs. Try to say at least how much can you get further with this. So try to divide and conquer the problem. And maybe even like if you reach a result, that's fantastic. Let's do it now. And if you need a break to drink some water or go to the restroom, now is a great time to do that as well. Okay. So how many people feel that they made significant progress in solving the challenge? Good. So I think this also gives it's a good example to take us into, remember that there is this thing called the imposter syndrome that we're often feeling, and probably you'll be feeling it in this lecture specifically, maybe in the course in general, because it's kind of like interactive, that you feel that like all the other people at Weizmann, or like in this, there's a good reason they were chosen for Weizmann, except for you, that, somehow are here by mistake, because in the interview you have like a nice smile, so they chose you to come into Weizmann, but you don't deserve to be here, and it would actually be found out probably next week when, they'll see that you don't know how to solve any of the exercises. But it turns out this is like a general phenomenon. There's lots of research about it. It's like, you can read it, in Wikipedia, on Google. It's called the imposter syndrome. It's like most people feel it, at least those that have critical thinking and are aware of the world. They tend to think that that's the situation. And it even doesn't change so much. Like it happens, to people in high school, and then it happens to them in their undergrad, in their graduate, in the PIs. It like somehow repeats itself. It's a psychological phenomena. What can you do about it? Be aware. It's good to be aware, and then you understand actually if other people are feeling it, so many of them, the majority, then maybe I'm okay, and you can continue in your life. What?
Not solving problems.
Not solving problems. Okay, so here I'm actually taking you to really a question. I wanted to give you the whole range of examples. if we started with the example of the massive online course where it's just like dividing two numbers, then you can pretty quickly, once you get the hang of it, infer all sorts of things. Then we moved into the beautiful drawing that I made. So you see a problem that's like, you see divider. Now we're moving to something that's kind of more complicated. Let me take you through all the steps. It's not something that I expect you, not in the first lesson. That's kind of like what I would expect you at the end of the course to develop more skill into doing. And if you did it now already, that's fantastic. Okay? So we'd like to, again, do some sort of a divide and conquer on this problem in order to make progress. So where would we start?
Size of a cell.
Size of a cell is a good place to start. So we'd like to start with size of cell. What would we do next?
Volume of the cell.
So from the size and volume is kind of the same to me. But
Mass of cell.
what?
Mass of cell.
So I can move to the mass of the cell. Good. What would be the next step?
Number of genes, proteins, and also.
Okay, so I would say there's different paths. I'll be showing you one path to do this, how to derive, and then we could actually do it in other ways. And actually, it's very good because I don't have time for all that, I'm sorry. But usually for everything you try to solve, you do it once, and then you try to do it in an independent way. It's also called a sanity check to see if the result that you got makes sense, so you want to do several ways. So what you suggested number is like another number of genes, etc. It's another approach, but let me run with that.
30% of water. This is something that we know from percent of water in the body, so we can take it to the cell and then we can subtract.
Great. So I want to, so excellent. So I want to go from the mass of the cell to the mass of proteins. And the way to do it probably would go through water and stuff like that. Okay? If I have the mass of proteins, what do I need to do next?
Mass of an average protein.
Yeah, so I need to know now the mass of one protein. And then I just need to combine these two lines, and then I get the number of proteins. Okay, we have a plan, as they say. We have a plan. Okay, you can see the divide and conquer now. It doesn't look so frightening. But, that's in retrospect, like, all the Nobel Prizes, when you read afterwards, say, not so complex, I could have done it myself, Not that I'm claiming, but just you see that, like, when you break it down, now, it makes more sense, but it doesn't mean it's that easy to do it. Okay, now let's try to do it. Okay, size of a cell. What do we know about the size of a cell? And like we're talking about bacterial cell, right? E. coli, these bacterial cells. 1 micron. Good. So really, a cell. The characteristic size about 1 micrometer. Okay, this symbol is like 10 to the minus 6. 1 micrometer. It's also the kind of thing that maybe you were not born knowing that the bacterial cell is 1 micrometer, but it has this nice property that it's not so complicated to remember. And once you remember it, you can infer lots of things. Again, this property of few to many, or like the power of combinatorics, if you want. That like with like 3 different numbers, I could infer 10 to the whatever properties, because it's all sorts of ways to combine them for different problems. So that's like really, if you don't know this number, now is a good time to etch it in your memory. Okay, so if it's 1 micrometer or 10 to the minus 6 meters, what do we know about the size of a cell? Where size of a cell, I'm thinking now of like the volume of the cell.
So it's the size of the cell.
Yeah, so it's this cubed, which is how much?
10 to the minus 18.
All right, so here it could be sometimes a bit confusing. So it's 10 to the minus 6 to the 3rd order. So that's 10 to the minus 18 meter cubed. Good. So I have this. Now I want to go from this to the mass of to the mass of a cell. Okay, good. So there's like, going from volume to mass, it's usually complicated. It depends. What's the density, this, that. but we're trying to make our life easy. Water, or like just, in general, a density of about 1. So how much would that be going from volume to mass?
10 to minus 12 grams.
It's 10 to 3 for meter squared.
Okay, so different people have different ways they do it in their head, and it's also very easy to get confused in these things. So it's usually good to take small steps, but each step you know what you're doing. Okay? So if like I have 10 to the minus 18, let's stay with 10 to the minus 18. I just want to replace this meter cubed, which is a meter by a meter by a meter. I want to replace it with like water or something of that sort, and to say how much would it weigh. So how much would 1 meter cube weigh?
1,000.
What was it?
1,000.
Yeah, so 1,000 kilos or 1 ton? Okay, 1 ton or 1,000 kilograms. Good. So it means that if I'm going from the volume to the mass, this would be now 10 to the minus 18 ton, because a meter by a meter by a meter of water is a ton, which is 10 to the minus 15 kilograms. How do I do that? Is it 10 to the minus 15 or 10 to the minus 21 or something else? It's 10 to the minus 15. I got it right. But if you want to be sure, there's a trick to do it. You multiply by 1. What do I mean? I multiply it by 10 to the 3 kilograms divided by 1 ton. How much is this? This is 1. So I'm always allowed, as a, to multiply by 1 is the one thing you're always allowed to do in life. So if I multiply by 1, okay, now it's good. Minus 18, 3 is 15. 1, minus 15 kilograms. Okay, which is also 10 to the minus 12 grams. How do I know? Same trick. Okay, so far so good. So I now have the mass of the cell. Now I want to go from the mass of a cell. By the way, how it also has these funny names, right? Like just like we had micro there. How much is 10 to the minus 12? Right, so there's these funny names like there's micro, which is 10 to the minus 6. Then you want to go down, it's nano, 10 to the minus 9. Pico, 10 to the minus 12. Afterwards, femto, 10 to the minus 15. After that, atto, 10 to the minus 18. Maybe there's below that. There's a zepto. Okay, we don't go there. I don't want to go there. Okay, so this is the mass of the cell. Let's go to the mass of the proteins. I'm sorry, it became a bit of a mess here. So let's do erase and conquer. Let's move it to here. Okay, I think you follow the path. I'm done with this. Okay, so this was mass of cell. Now we have mass of proteins. How would we go from 10 to the minus 12 to the mass of proteins? Again? Who suggested something? Yes? 1/10. I love it. Why 1/10? We love 10. So this is like would be 10 to the minus 1, or 10% of the mass would be proteins. I like it very much. Can we do a sanity check to see if it makes sense or not?
30% should be upper limit because of the water in body.
Okay, so that's like one factoid that we know from, I don't know, school or something. We're 70% H2O. So we left just with 30% of all the rest. What is the rest? Fats, lipids, which is the same, DNA, carbohydrates, proteins. Okay, so let's make, without making a big deal out of it, let's say half is this, half is protein, half is the rest or whatever. Half of this is about 10%, something like that. Okay, so I have 10 to the minus 12 grams, so now I have 10 to the minus 13 grams. Right? Okay, now I need to do like, if it was gymnastics, now I need to do like some really clever trick because I need to do something to go. All of this is like what's known as the macro scale. I'm looking at like weights and size and things that we're used to. Meters, this MKS units, blah, blah. Now I want to get into the logic of the cell. Why? Because I need to combine it now with the mass of one protein. Anybody know something about the mass of a protein? Yeah?
We can go from proton, from the weight of one proton, and then we know that there is 10 protons in one atom, and then you can, and there is like probably like, I don't know, like 100 atoms in one protein.
Okay, so we can try and build it up from there. It's not easy to go directly just from, because the question, how many protons are in a protein, it's 10 to the not so clear immediately how much. So it's a good direction from the base here, but it's pretty tough. What are you saying? Yeah.
I mean, we know that proteins are usually on the mass scale of kilodaltons and up a little bit, depending. So we could do kilodaltons times 10 to the 2 or something like that.
Okay, so those who come from biology, more biochemistry, biology training know there's this thing called kilodaltons. Daltons are the units of exactly like protons. Kilo is 10 to the 3. And now you need, it's like in these kilodaltons something, the mass of protein. So that's one way to approach it, yes?
Building on that, you know the average size of a protein in amino acids. And you know the average weight of an amino acid in daltons, which is grams per mole. And you know the average amount of space that a mole is. And you can multiply all of that together and you get the weight.
Okay, so the suggestion here is to talk about what we know about the protein, which again, you need some information, and you see it's the sort of thing that it's true. It's deriving this, you can't do it just, you woke up in the morning, I don't know anything about the world, or you can try, but it's kind of tough. So you need to know something. For example, if you know something about a protein, how is it built, in terms of like how many amino acids it has, and what's the mass of each amino acid. We could talk about it, we don't have a lot of time, so I won't do the derivation also that way. Anything else that people wanted to suggest? Okay, so how many have heard about, kilodaltons and all that in terms of units? How many, not until now? Okay, good. So let me think how best to proceed from here. Okay, so let's say we did this jump of logic that we know that it's, say, around 10 kilodalton. Okay, I'm trying to take like that. So we'll talk about it in the next class when we'll start talking about sizes and concentrations and things like that. We could see how do we derive these things from like much more basic principles. But so just to remind ourselves, Dalton is like the mass of like a hydrogen atom or a proton or something like that. So that's very useful when you go into the molecular world. Okay, and that would mean like 10,000. Okay, actually it's somewhat more, as we'll talk about. It's like 30,000 or something like that. So, actually there's a nice way to do that, and that is to write it as few times 10 to the 4 Dalton. Okay, what did I do here? I again using something a bit strange. For the kilodalton, I'm having like 10 to the 4, it's like 10 kilodalton, but it's a few times that. What is a few? A few is like 3, okay? Why? Because let's put this an aside. Let me show you something kind of, I think, neat. There's like a new type of algebra we could do, which is 1 few 10. Okay? Let's see how much. Let's see if we got it. So in few, we could just write as f to make it even simpler. Okay? So how much is f times f? 10. Okay, and with that, you'll see we could do all sorts of useful things. Okay, I'm saying all that because I want to move forward. So it's a few tens of kilodaltons. Good. So now I have a problem. I have the mass in grams, and I have this in these units of Dalton. How am I going to combine this? And it's actually, this is like, again, it's a complicated lesson today. but these things that would recur again and again, so once you get them, you're in good shape. So this is like the macro scale, and this is the cell biology, biochemistry, molecular, microscopic, of like things measured in Dalton. Now we need to do the conversion between Daltons and grams. Okay? We basically need to know how many Daltons are in a gram. So we're not the first people to do it. There's this guy, Avogadro, that did it before us. Maybe you remember meeting him at some course. But let's try to derive it as well. We could try very quickly. So what do we want to know? We have a gram filled with stuff, material, whatever. And we want to know how many Daltons would be sitting in this 1 gram. Or not one, that's what we need to know. Okay, how can we do it? Let's do the following. Let's say I have a cube of 1 meter, and I'm filling it with stuff. Stuff is made out of atoms, and then from that I want to infer, okay, so atoms, what would be the, atoms is great, because atoms are in these units of Daltons. Okay, so how would we go, what's the size of an atom?
1 angstrom.
So there's this unit 1 angstrom, which is really the size of an atom. That's also something useful to carry in your head. So an atom is 1, they write it this way, angstrom, which is 10 to the minus 10 meters. Okay, as I said, a lot to learn today. Forget the imposter syndrome. You came here to study. Take whatever you can. Okay, so we have 10 to the minus 10. That's the size. So how many would be sitting inside this box? How many such atoms could sit inside? So 10 to the minus 10 to, so I could put 10 to the 10 here, times 10 to the 10, times 10 to the 10. Okay, so the number of atoms is roughly 10 to the 10 cubed, which is 10 to the 30. Okay? So I have 10 to the 30 Daltons per meter cubed. Okay? Good. So a meter cube is like a ton, right? So now I want to go into a kilogram, or that's a liter. Or okay, so that means that if I want to go from meters cubed to a kilogram, that would be 10 to the 27 Daltons per kilogram. And if I want to go to a gram, which is what I was looking for, it's 10 to the 24 Daltons per gram. Which what? And so I have 10 to the 24 Dalton, 1 gram. What we just repeated is what took Dalton or Avogadro many, many years. Okay, somebody remembers what's the Avogadro number? 6 times 10 to the 23. 6 point something, something, something. Okay, so 6 to the 23, for me 10 to the 24 is kind of like the same. Okay, so we kind of derived this Avogadro number out of thin air, which I think is neat. If you don't like this, maybe you're in the wrong course. No, I'm not. This is not like it's actually also like it's physicsy and biology and everything together. So I'm not saying it's. Anyway, so 10 to the 24, good. So let's try and put it all together. You remember we were here in 10 to the minus 13 grams of proteins per E. coli cell. Now I just want to multiply it again by 1. Why? Because I want to go from the macroscopic to the microscopic world, so I'll multiply by 10 to the 24 Dalton per 1 gram. You see what I'm doing? It's this nice unit conversion, which usually has a bad name, like it's not interesting unit conversion, which is true. It's not so interesting. But at the same time, it's like a really essential tool, and you have to do it right. Otherwise, you're like, in a mess, or you just can't do anything. Okay, so that brought us to minus 24 minus 13. That's 11, right? So I have 10 to the 11 Dalton of proteins inside the cell. Okay, let's bear with that for just a minute and go back to our worry about the course. let's talk about mathematics. Okay, one thing I know that people are often very worried about going to a course, on say quantitative thinking, etc. That some of us like math more, some of us feel less comfortable with that. What I think you'll find that in the course, but also I think in general in the kind of quantitative reasoning that is needed, if you are okay in junior high school in terms of math, you'll probably be okay here as well, because we don't need much more than what was used in high school or junior high school often. We just multiply and divide, and we need to know 10 to the something, and that will take you a very long way. So I think, but if you're good at that, doesn't mean you're good at quantitative reasoning, right? The logic doesn't go in the reverse. There's a lot of things that you don't need to know math very sophisticated, but the simple math, you have to use it in a very well-informed way. Okay, that was an aside. We have 10 to the 11 Dalton. We said each one is f times 10 to the 4 Daltons. So now I just need to remember to combine these two together, divide this by that. So I have 10 to the 11 Dalton divided by few times 10 to the 4 Daltons. That's equal to what? I can take this 10 to the 11, it's like 10 times 10 to the 10. 10 divided by few would be few. 10 to the 10, 10 to the 4, 10 to the 6. Okay, so I got a few millions, which if we go back here, it brings up to this answer, which interestingly, so I did it pretty quickly and roughly, but it turns out that in almost all organisms, not almost, like in all organisms that I'm aware of, all cells, you have something like between 2 to 4 million proteins per micrometer cubed. So here's a rule of thumb if you don't want to repeat this calculation every time, or you can just repeat it. But actually, so we asked a specific question about proteins inside E. coli. But what we arrive at is actually useful for E. coli, if it's 1 micrometer cubed. But actually, it could be generalized. It's true for human cells, for mice cells, for plant cells, for viruses, kind of. That it's actually pretty general in terms of what you infer afterwards. So that's also a nice thing about using numbers in this sort of way. Also, in trying to look for these sort of answers that you get something, you see what is the answer here. It's also kind of like different than what we used to, right? Usually in the things that you did in your school and then in undergrad and then in your research, you're not expected to give an answer of 10 to the 5 to 10 to the 7. Usually they ask you, okay, give me 3 digits. But it's, I don't want to say bullshit, but it's not really how answers usually look in the real world. In the real world, very often, you don't know anything very accurately because it's ugly, complicated. There's all sorts of things coming in. And you just need rough answers like this, or maybe if you then can go to even more general rules of thumb, that's great. And that often happens, like you came from a specific question on a specific cell, but now you can know about all of biology, something about concentrations of proteins. Where are we standing? So we talked about this, we talked about that, we talked about this, and about this, and we talked about units, and we did this algebra, and we talked about the fact that answers and generalizations. I think we're making progress here. Yes?
Is this like in a natural state? This rule of thumb, is it for in a natural state, or is it like the physical capacity of a cell to hold that much protein?
So that's the physiological state of, I think, all of living life that I'm aware of. Again, viruses could be a bit more, crystallized, whatever. Where does it come from? Is it because of a physical limit or something else? That's a great question. That I think that's the sort of thing that I want to say. Like, new question, once you take this perspective, now you can ask, can it be much higher? Can it be much lower? What do you think? Just 1 second. What do you think now? So now it's pretty interesting, right? Because the way we derived it, now we have answers to some of those deep questions also. It can't be much higher, right? Because there's just no way. Like it's like, and if you want some water, etc., it's like really kind of like at the top of what it could be. And if you want some other stuff, which then suggests, okay, could it be much lower? Maybe, but it seems like then it might be, I don't know, something with could also be explanations. Is it fitness, etc.? Okay. There was somebody else that wanted to say something about this? We're good. Any more comments or questions about this? Okay, so we have a few more minutes. So now let's step back for a second. We went through quite a lot already. Now let's talk about some of the way this course would work from now onward. Okay. I leave this here? Yes. So first of all, look here. Everything that will be, you want to follow about the course, etc. We have the website of the course, weitzman.acil/cbn, cell biology by the numbers. All the details, etc. you can find there. For example, you can find the next exercise. So I think, it's nice to come to a show and like participate somewhat in but really the way the learning happens is also by, fighting against some specific questions yourself. And for that, every week we'll have an exercise. Which is already available online. Not yet. It will be available in an hour. And, but from then on, it will be available forever. And
You're not using the Moodle.
What?
Also uploaded to the Moodle, or you're not using the Moodle?
We're using the website, the course website. I hope that's enough. If that's OK, if there'll be any problems, let us know. So the exercise you see is composed of several questions. I think it's, if you don't do that, you're not really practicing, so you should do it. The exercise should be handed by the start of the next lesson. So you have a week to think about it, look at it, and hand it over. I'm right or am I mistaken? Yeah. So for next week, you already need to hand in the first exercise.
And then it's sending to this mail, right?
Okay. Right. So this was the website. Now about this is the email of the course. So cellbiologybythenumbers@gmail.com. That's our way to communicate in any way you want. So everything that you want to hand in, you just send to this website, to this email. If you have any questions, feel free to send there as well. If you have, somehow you can't do it this week, there's a problem, something happened, a family, too much load, we understand. That's fine, just write us. Okay, say I need another something, we'll make it happen. Okay, we're pretty flexible, just let us know so we understand where things are standing. For example, if you want to leave the course also, say that's fine, so that we could just follow what's happening. The people who would really help me make this course work are the two teaching fellows, Ron Sender and Leon Greenspoon. I think it would be nice if you present yourself. So come over.
Hello. So I'm also Ron. It's a necessary thing if you want to be in our lab, to be called Ron. I'm just finishing my PhD at Ron lab, and I'm working on this kind of by the numbers approach, like related to the human body and the human cells. And I will present some of my early work soon, so hope it will be interesting for you.
I think, yeah, you can clap.
So hi, my name is Lior, not Ron. I'm also a PhD student in the lab, a second-year PhD student, and I'm working on using this like by the numbers approach for quantifying like wildlife and specifically wild mammals. So we're also going to talk about it during the course, but if you're interested in talking about it before, feel free. I will say for me and Ron that we're in the Benozio building, 6th floor. So if you also want to like chat or like please come find us, we'll be super interested to do that.
So in general, I think you'll find in the course that you'll have to work hard, and we'll also have to work hard. So I think we should, all respect each other, try to, be aware that, it might take us some time, etc. But also, I don't know, just, in general, we'll try to give you the best service, to try and improve your capabilities. and we're expecting some things. That's the kind of, if that's too much for you this year, you can come and do it next year. That's completely fine as well.
And for the assignments, are we allowed to work in groups, or like how does it work for you guys?
So these assignment, they're made for working, individually. I think it's completely fine if, after you made your effort to, to fight against the exercise individually, you find that, you're stuck with something, you want to consult somebody, that's fine. But, in that spirit. More questions?
Is it possible to give an estimation for the workload each assignment should take from us?
10, I would say.
10.
I think it's up to a few hours. It's not a lot, and there are about 6 exercises.
So between 1 and 9, you're saying.
Yeah.
Okay, so there's gonna be, we have a weekly assignment, but as you'll see, we won't have, there wouldn't be lectures in all the weeks, or some of the weeks would be kind of special lectures, as you'll see. So altogether, I think we have 6 exercises. Yeah, so 6 exercises. For each exercise, the workload depends also on you, but it's a few hours. Okay, if you're working more than 10 hours on an exercise, let us know, come consult, maybe we could help you or somebody else. So it's supposed to be less than that. But if you're finishing it in half an hour, I'll be surprised. So it's something, What is the teaser? We'll get to that. That's a good question. We'll get to that in a second. Okay, grading and attendance. Okay, so in terms of attendance, we're expecting you to attend the class. Why? I hope you've been here and now you experience, that's the kind of style I think the teaching should work, so you should be attending. At the same time, we're aware that sometimes you can't attend, once in a blue moon, as they say. So if there's a reason why you can't come, write us an email to here and let us know I can't come this week or I can't come next week or something like that. And again, we understand. We just want to be aware and to be able to follow this. There will be recordings of the course. There's already recordings of previous years, but I think this, we'll see exactly how long does it take, but I believe pretty soon we'll have this recording also available online. We'll have a link from the website of the course. And so if you have friends that missed the first lesson and you think that's the best course they could take at Weizmann, you could tell them, come next time, look at the exercise, look at the video. You could still join, no problem. And again, if you'll be missing a lesson, we'll try to make it available in that way. Okay, grading. So half of the grade is based on the fact that you're alive, attended the course, came and participated, and handed the exercises. Okay, we won't be, in the exercises, we won't give you, exact grades. We'll try to do a lot of work in order to give you comments, constructive comments. But if you'll be doing your exercises and you'll be attending, you'll have half of your grade done already. More than that, if you'll have really super smart things that would surprise us in a positive way in the exercises, you'll get a bonus. Special bonus. We'll see how it goes. That would be half of the grade. Then at the end of the course, which as you'll see, why is it 6? Because actually we're doing it such that 3 or 4 weeks before the end of the semester, we're finishing the lectures in this format and we're moving into presentations of the final exercises. Final exercises is each one of you, believe it or not, will be standing instead of me in front of a blank whiteboard, and you'll be presenting in 5 minutes a problem that you found interesting because it's something you do in your research, because it's something you're curious, because you have a hobby, because of something else, and you'll be writing the question and doing some calculation to try and get some insight into that. Okay? I don't know. Previous year, some people did it on whether you can diet, whether you can have a diet based on drinking a lot of ice-cold water, that you'll be warming, you'll be taking heat and energy and calories from your body in order to warm the water, and whether this could be the basis of a calorie. Okay? Or if you're traveling on the train from Tel Aviv to Rehovot with somebody who has a flu, how many virions are you gonna inhale? And many, many other examples. So it could go in many, many different directions. But it's a place to put your creativity and your interest, etc., and you'll be doing it. And it's actually, I think, also a learning experience in terms of that. But in order to be able to do that, you'll see we'll have a method where you'll have like a mentor inside the group that will help you. You'll be practicing. There's this, there's that. Anyway, so that's another 25% of your grade. And the last 25% is on this thing that you presented in class, you'll be handing in a vignette. What is a vignette? A vignette is like a short story, or kind of like the calculation, which is a question and some answer. I really like vignettes so much that I wrote a whole book about it. So this is the book, which is also the book of the course. It's called Cell Biology by the Numbers, although now we're moving into mammals and other stuff and things in sustainability. But this is based on different questions. So you can pass around the other copy. So what you'll be doing in the final exercise, kind of like there's like 90-something examples here in the book. You'll be going through them in some of the exercises. So you'll be doing something similar. It's like a 2-page thing where you raise a question. Let me read some questions. Just how many cells are there in an organism? How big are genomes? What is faster, transcription or translation? How fast do cells move? How much energy is released in ATP hydrolysis? All sorts of questions like that. And you'll be handing in a 2-3-page write-up with some numbers and something that would give some insights into that. Okay, and that would be another 25% of your grading. Okay, so I think we got to 100%, right? So 1/2, 1/4, 1/4. That's the idea. Bonuses if you did something that puts us at awe and that we really like.
The vignette is another topic?
So usually it's the same. No, not usually. It's the same topic. It's what you presented. Also, we don't want to burden you with so much work. We know it's going to be quite some work anyway. So yeah, usually it's like what you presented in class. Now you also do it in writing. It's usually good to start writing it before. Also, we're doing like some mentoring, so people give you insight on whether it's clear and how to improve that. And I think it mimics also the processes of like both giving a presentation or giving a elevator pitch and writing a paper or writing a grant proposal. So we're trying to develop also all those skills at the same time. Okay, we have that. Any more thoughts, questions about the course, the method? I'm listening. Okay, I definitely created some build-up. Okay, so last thing, in preparation for next week, let me ask you a question. What is no, let's make it. What is heavier? An mRNA or the protein it codes for, no shouting please, A, the protein, B, the mRNA, C, roughly the same, or D, it depends. Okay, so just to clarify what's the question. So I guess, biology, the central dogma, this fantastic thing that, we might be taking for granted, but when you think deeply about it, it's like really amazing. We have DNA. Out of that, we're reading messages. We're writing them as like these short emails called mRNAs. And then we're reading those emails into machines called proteins. And now the question is, what you can keep on moving the book. I don't know if everybody saw that. What weighs more, the mRNA or the protein it codes for? Okay, fair question. I'm putting on that. And we're asking what it's the protein. Okay, who votes for A, the protein?
Just clarify what organism you're talking about.
Yeah, so which organism do you like most?
Just multicellular or unicellular is the question.
Dog. Okay, we're talking about dogs. I just heard we're talking about dogs. Okay. Who votes for the protein weighs more? I have about 10 people. Good. Who votes for mRNA weighs more? I have about 20 people. Who votes for roughly the same? I have about 10 people. Who votes for it depends? Another 10 people. Okay. I think we have a good distribution. So we'll have something to talk about next week as well. So we have I think you know what you need to do now. You need to, if you're not signed, some problem in your, come and talk with us. If you are, go and check for the exercise in like an hour. Be ready to hand it in before the next lecture. If you can't come to the lecture, you can't hand in the exercise, you want to do something else, shoot us an email. If I forgot something, let me know now, or be silent forever. We're good? You want to say something? Yeah, right. Okay, one thing that I forgot. So obviously, so if you are not here, you are here. So you can't not be here. But in the next few lectures, maybe you won't be here. Our request is the following. Will be, as we said, the lectures will be available either on the website or on YouTube, etc. You need to watch them. And you need to write us an email there of two things. One is something you took out of this lecture that you just watched. And the second is something you think could be improved in what you just watched. Okay? Clear? Yes.
Can you put all this information just on the Moodle so that if anybody didn't take a picture of this or forgets it, like just so we will.
Yes. Okay, I know some people have to go. Anything else? Thanks for coming. Have a great week.